All right, today we're going to dive into a microbiology test that's been around for over 100 years. It looks simple, right? A couple of test tubes. But believe me, inside this little tube is an absolutely fascinating story of survival, adaptation, and even evolution. Let's see what it has to teach us.
And it all boils down to this stark, unmistakable change from a deep forest green to a brilliant Prussian blue. I mean, this isn't just a pretty color change, it's a verdict. It's a clear-cut decision that tells us whether a tiny organism can pull off an incredible metabolic trick.
So what's really going on here? How can this one simple reaction tell us so much about a bacterium's identity, its capabilities, and even its secret genetic playbook? Well, that's the exact puzzle we're going to solve together. Okay, so let's jump back in time a bit. The citrate test was first dreamed up back in the 1920s for a really important reason. They needed a way to tell the difference between bacteria that signal fecal contamination in water, like E. coli, and other harmless bacteria just hanging out in the environment. It was, and honestly still is, a critical tool for making sure our water is safe. The first version of this test, created by Stuart Koser, was a liquid broth, and it had this one major flaw. Any kind of cloudiness, even if it was just from dead cells floating around, could trick you into thinking you had a positive result. But then, just 3 years later, a guy named James Simmons comes along and has this brilliant idea. He adds agar to make the medium solid, and this is the game changer. He adds a pH indicator. And just like that, he turned a subjective test into the crystal clear color-based standard we still rely on today. Okay, but here's where it gets really cool. The way this test actually works. The medium itself, this Simmons citrate agar, it's not some cozy, nourishing soup for bacteria. Oh no. It's an extreme survival challenge. It's a metabolic gauntlet, specifically designed to weed out the weak. The test basically creates what scientists call a nutritional bottleneck. It's like throwing a bacterium into a locked room with only one type of food on the table and saying, all right, can you live on this and only this? It is a pass-fail exam for a very, very specific skill. And wow, this slide really drives home just how brutal that environment is. I mean, look at this recipe. There is one and only one source for carbon atoms, sodium citrate. There is one and only one source for nitrogen, ammonium phosphate. Everything else is just there to keep things stable. And then you've got our little spy, bromothymol blue, the pH indicator, just waiting for the signal. So you get it now, right? To survive this gauntlet and turn that tube blue, a bacterium needs a very specific set of genetic tools. It has to have a secret playbook for using citrate that, frankly, most bacteria just don't have. So how do they pull it off? Well, it's a two-step plan, really. First, the bacterium has to have a special gate on its surface, a protein called citrate permease, to physically pull the citrate inside. That's step one. Then, once it's inside, it needs a kind of molecular scissor, an enzyme called citrate lyase, to chop that citrate up for energy. And it's what happens after that step that gives us the big reveal. See, as these little guys are munching away on the citrate and the ammonium phosphate, they start producing alkaline waste products, stuff like ammonia and carbonates. This waste starts to build up in the agar, right around them, and that makes the pH go up. And when that pH crosses the magic threshold of 7.6, our little spy, bromothymol blue, flips its chemical switch, and bam, the whole tube turns that gorgeous blue. And just like that, this simple color change lets us sort bacteria into two totally different teams, the ones that can do it and the ones that can't. So let's meet the usual suspects. All right, first up, you've got the citrate all-stars. These are the ones that turn the tube blue. We're talking about bacteria like Klebsiella, Enterobacter, even some species of Salmonella. All of these guys have the complete genetic toolkit. They've got the gate and the scissors. But what about the other team? Well, the most famous player on the citrate negative side is good old Escherichia coli. And this quote right here explains the really fascinating reason it fails the test. It's not that it doesn't know how to eat citrate. The problem is that it can't get it in the door when oxygen is around. Think of it like this. E. coli has the molecular scissors, but it doesn't have the key to open the gate. The gene that builds that gate is automatically switched off anytime there's oxygen present. And since we do this test in a regular old test tube with plenty of air, that gate stays locked shut. The E. coli is surrounded by food, but it starves, and the tube stays green. And for decades, this was microbiology gospel. E. coli is citrate negative, period. End of story. But then an incredible long-term experiment came along and showed us that even the most fundamental rules of biology can be rewritten by evolution. I want you to just look at this number for a second. 31,500. That's about how many generations it took for a population of E. coli in Dr. Richard Lenski's lab to do something impossible. To put that in perspective for you, we're talking about more than 15 years of continuous daily experiments. So picture this. For over a decade, scientists had 12 identical populations of E. coli growing in flasks that were full of citrate. And for years, they all ignored it. But then, one day, the scientists noticed something wild. One of the 12 flasks had become incredibly cloudy. That one population had spontaneously evolved the ability to eat citrate, even with oxygen all around it. So how in the world did it pull this off? It was like an evolutionary heist in 3 acts. First, some earlier unrelated mutations had already made the bacteria's DNA a little unstable, kind of priming the pump for a big change. Then came the main event, a random genetic copy-paste error. This duplication accidentally plopped the gene for the silent citrate gate right next to a genetic on switch that's always active when there's oxygen. This basically hotwired the gate, forcing it to stay open. And finally, over time, more mutations fine-tuned this new ability, making them super efficient citrate eaters. Okay, so we've gone from a simple color change all the way to watching evolution happen in real time in a flask. After all that, what can this humble little test tube really teach us? Well, for one, it shows us that a really clever, simple design can uncover incredibly complex secrets about how cells work. It's also a great reminder that this test, even after 100 years, is still a vital tool we use every day. But, maybe the biggest lesson is that the rules of biology aren't always set in stone. Evolution is a real, creative force that we can actually watch happen right before our eyes. So yeah, the next time you see this test, remember, it's so much more than just a diagnostic tool. It's a tiny arena for huge microscopic drama, a story about the challenges of life, the amazing genetic solutions organisms come up with, and the unstoppable engine of evolution itself. And that just leaves you wondering, right? That was just one gene in one bacterium in one lab flask. It makes you think, what other incredible abilities are lying dormant right now in the genomes of the trillions of microbes all around us, just waiting for one lucky mutation to change the world?